First, I want to apologize for being a little bit late. We got lost, which is very typical situation for the biologists. You can get You can really get lost in the amounts of biological data, and I will tell you more about it during my talk. And what I will be talking about is integrated bioinformatics systems and tools that we are developing at Bioinformatics Group at Computational Institute of the University of Chicago and Mathematics and Computer Science Division at Argonne National Lab. There is a lot of people working in this group, and they're absolutely wonderful, and along the talk you will understand what exactly we are doing by doing that. So what are we going to talk about? First, I will talk a little bit about our approach to study evolution of genomes, metabolic networks, protein families, and many more. And this is about bioinformatics, which is absolutely a fascinating subject, which promises a lot, delivers quite a bit, and probably will change this civilization in the future. So then I will tell you a little bit what are we doing. We are building the extra large information systems for high throughput analysis of genomes and metabolic reconstructions for public genomes and user submitted genomes. And if we don't have the tools to analyze this data, we just build them. I will also talk about grid-based integrated computational infrastructure, because the analyses are so high throughput, so complex, so tedious, that it will be just inhuman for people to do it manually. So we are automating most of the processes that we are doing. But the most interesting part to me, because I've heard myself giving this presentation many, many times, so this presentation is not interesting to me, but the part that is really interesting to me is the problems. And we do have a lot of problems in bioinformatics, and we do need help in solving of these problems. So let's start. The last several years, not more than a decade, there was a madness in sequencing of the genomes. There are so many. Before I will go any further, do you have any background in biology? Do you know what sequencing of the genomes is? So in this case, I will just talk your language. So there was a lot of sequencing of the genomes, and this is sheer madness. What drives this madness is a lot of different groups are interested in sequences and genomes of particular organisms. But what emerges from this variety of the organisms and individual interests of individual groups is an ability to look at the data and do the comparative analysis across multiple evolutionary domains to do efficient comparative analysis, because what is interesting is that the major approach that bioinformatics takes is the comparison. It's a comparative analysis. It's a science of big numbers. It thrives on the high throughput comparisons of what is known to what is unknown. So in this case, you can transfer annotations by similarity from what is unknown, from what is known to what is unknown. You can compare functional and genomic properties of different organisms from different taxonomic groups. Like, for example, what is the difference between the sequences, and what is the difference between the metabolic pathways for the organisms who live in thermophilic environment versus the organisms who live in Antarctica? There should be the differences, and these differences can be analyzed and formalized on the level of the genomic sequence analysis. Or, for example, what is the difference between the pathogenic organisms and non-pathogenic organisms? Just to give you a small example, for example, the BCG strain of mycobacteria, which is used for vaccination against tuberculosis, is different from the pathogenic Mycoplasma tuberculosis only in 13 genes. And by comparisons, you can probably identify pathogenic factors that are directly connected to the disease. So in this case, it's obvious that more data you have, more diverse the set of genes that you will be looking at, more interesting patterns you can identify, more interesting comparisons you can do, and more interesting information you can derive for bioengineering, genetics, medicine. So The goal of this madness is actually to make the efficient information base for deriving the conclusions about the organism's functionality. What is the same and what is different in different biological organisms, and how it affects function. But also what I want you to note on this slide is geometrical progression of the data. And I will return back to this geometrical progression of the data later in my talk in the problems side. So we decided that the major thing for us and the major emphasis is comparative analysis. But in order to support this comparative analysis, what do we need to do? First, biological systems, they are very complex. It's not only the sequences. Sequences have, like DNA is followed by the gene products. Gene products perform functions. Functions are connected into the complicated networks of interactions. So we need to understand the functionality of biological systems so we can somehow control it and modify it. We need to understand functionality of the systems on various levels of organization. And that's why we need to integrate information from sequence databases, features databases, metabolic databases, regulatory databases. More information you have, more different angles you can look at this information, more comparisons you can do, better you will understand the functionality of the system. So integration of the data is one of the major requirements for efficient bioinformatics analysis. The second one is we need to have tools and algorithms for pattern recognitions and comparisons of biosystems on various levels of organization. It's just the comparisons of the sequences is way not enough for contemporary bioinformatics. We need to push it much further. And this is, I'm also going to talk about it in the problems part of the talk. We also need, but because the amounts of data are so humongous, we need scalable computational resources to do this across-the-board comparisons. We need to have on-demand scalable systems. And in this case, we really need to have heavy-duty computational backend. So the good news is that we can do it now. Sort of, sometimes better, sometimes worse, but we can do it now. So let me tell you a little bit about our approach to understanding of the biological systems. We firmly believe that to understand biological system is possible if you will understand the way it was evolving. Because what was happening during the evolution, it was almost like genetic engineering in action. The systems were, the nature was tweaking the system to adjust to different the environmental conditions. It was modifying it as much as needed for the organism to survive. So in this case, if you will look from the evolutionary perspective, then you can understand the logic of the changes. You can understand why these components were changed and how they were changed. But what is happening is that nothing is changing in isolations. Like, for example, everything in the biological system co-evolves. That's why it's a system. It has a systems response. You have a signal, you have the systems response, which means that like, for example, protein families and the genomes, they are changing because metabolic networks are changing. And metabolic networks are changing because the environmental conditions are changing. Like, for example, if you will pour sulfuric acid onto the organism, it's better to do something. But exactly what's needed for survival under this harsh conditions. So pretty much everything is co-evolving. So if we want to look just at the protein families, we will never, ever understand the logic of the changes. So, and what forms these changes is environmental conditions. Something changes in the environment, you better mutate. Taxonomy. Taxonomy imposes constraints because you can't evolve without constraints. Like, if I am a human, I will be evolving like a human. If I am an elephant, I will be evolving like an elephant. So, if you are an archaebacteria, you have certain commitments, you have certain history, and you have the boundaries of changes. So all archaebacteria will share certain properties. So Also, physiological profiles. There are major design commitments that organisms are making. Like, for example, to breathe, to use oxygen for respiration or not to use oxygen for respiration. They are major building blocks, major decisions. Like, for example, Teilhard de Chardin, which was a wonderful evolutionarist at the beginning of the 20th century, he thought that evolutionary changes is everything that is related to the new access to the new energy sources or access to the new kinds of information. So this is evolutionary changes. There is a lot of diversity, but really big design commitments, what energy to use, or what sources, mechanisms for acquiring of information to use. This is evolution. So having all this in mind, we started to build the integrated information system. And my group built the Puma 2 NER system to provide an environment for co-evolutionary, as we decided, and comparative analysis of genomes, metabolic networks, and enzymes in the framework of taxonomic, phenotypic, and physiological information. And the reason why we did it, because we really want to understand the logic of the organism. Without understanding of the logic of the organism, it will be very difficult to interpret the events of molecular evolution. So what Puma has now? Puma is an interactive, integrated environment for high-throughput genetic sequence analysis and metabolic reconstructions with grid-based computational backend. Every single word in this definition means something. And I will tell you a little bit more what every single word in this means. And these are the screenshots from the Puma system. So it integrates information from over 25 genomic, metabolic, structural, and taxonomic databases. We are taking all sequence data that is available in the public domain from NCBI, UniProt, PIR, and we're integrating it in one environment. Then we are also integrating information from the available metabolic databases. And, for example, structural databases, PDB, and so whatever we can get hold on the information we're integrating and we're warehousing it. And I'm repeating this, we're warehousing it instead of doing something more reasonable. And I will complain in the last part of my talk where we're warehousing it. But it's not enough. This information is not enough for us. So we decide to amplify this information. So we are taking all of the sequence data that is available, and we are actually analyzing it. And the grid technology allows us to analyze it using BLAST, blocks, interpro. So we're amplifying the data. We're annotating it with additional features. And the reason why we are doing this, because we want to do the comparisons more efficiently. We want to recognize patterns more efficiently. And that's why we're amplifying the data. So we are doing automated metabolic reconstructions. And I will tell a little bit more about what does it mean to do the metabolic reconstruction. But in a nutshell, if you have the functions of the genes predicted in the genome, then you can superimpose it on the functional networks from the databases like Cargo. We have our database EMP with metabolic pathways. And without knowing anything about the lifestyle of organisms or their physiology, you can actually reconstruct the physiological profiles. You can reconstruct the features that organisms have and predict certain features just from the genomic repertoire. It will be sure enough, it will be not complete, sure enough, it will have some of the pathways, some of the genes, they will not be expressed, but at least it's a valid model to start from for the experimentalists. So we have a suite of unique tools for evolutionary analysis of enzymes and metabolic networks. I told you, we don't have tools, we don't wait, we just go ahead and build them. And we have satellite databases. The satellite databases, because we want to look at the certain parts of the biological story in more details, like for example, and also because some people are paying us money to do that. We're part of 3 biodefense NIH centers of excellence, and that's why we are creating the PATHAS database. And we're part of the structural biology, NIH Structural Biology Center, and we're predicting biologically interesting targets. So we have several different refined data sets. So the interesting system about which I want to complain in the last part of my talk, I'm promising so much complaints, but I will really do, that this is NAIR system for high throughput analysis of user-submitted genomes. If we have this madness, if we have this sequencing madness, which comes as an avalanche of data, you better have automated systems that will allow the user to download your genome, and it will be automatically annotated, and then you need to provide the interactive environment for the users to actually refine the analysis manually. But you need to start from valid, good, automated model for genome analysis. And so far we have, without any publication yet, we didn't even publish the paper, but we already have quite a bit of users. And like we have Yersinia pestis and Haemophilus influenza, 30 strains of Haemophilus influenza, and we have Chivanello Federation from DOE is using us. So we have a lot of different things. We can process and fully analyze, and I will tell you what does it mean to analyze, the bacterial genome in 4 hours. And it performs the analysis and displays the results. So what are the steps of analysis that PUMA does and NAIR does? First, assignment of functions to the genes and characterization of the proteins. So once users have submitted the genome, we are running it on the grid against the array of tools, including BLAST, as I said, BLAST, blocks, TMHMM, PFM, our own tools, Chisel, and we are collecting all this information to input it into the voting algorithm, which will vote to predict the functions of the genes. So it's not enough just to run BLAST on the sequence data to assign functions to the genes, because it's too crude. We are collecting information about domains and active sites and whatever we can get in order to assign functions to the genes reliably. So then what we are doing, we are superimposing it on the collection of metabolic and functional networks. The assigned functions, we are superimposing them onto the known pathways. And this technology was developed by Evgeny Silkov in 1993. And so we're predicting metabolic pathways and providing it an interactive environment for users to refine. And then my favorite part comes, how to predict the phenotypes, how to predict the physiological properties of the organisms, how to identify evolutionary and co-evolutionary patterns and signatures. And to do that, we need very efficient ways to do the pattern recognition, and it's better to be multidimensional pattern recognition. We don't have a lot of algorithms that we desperately need, and it should be all in comparative framework. So this is how sequence analysis is done and presented to the user in PUMA and NAIR. So you have information from public databases integrated in the header. You have pre-computed data on the right side of the slide, and it shows the similarity searches. This is blocks, this is interpro, domain analysis, BLAST. If you will go down, there will be BLAST against PDB structural databases. If this information is not enough for the user, on the left side he can do interactive analysis. So there is a lot of possibilities to refine the analysis, and the user can add comments and annotations. this is how the metabolic reconstructions look like. And what is happening is, this is, for example, the pathway from the EMP database. We can also do the reconstruction from the KEGG database. And we need to have an engine how to add the user-submitted pathways. And what it gives us, like, for example, this pathway, it represents the glycolysis, which has an alternative behavior. Some of the organisms, they perform ATP-dependent, they have ATP-dependent phosphofructokinase. The other organisms, they have pyrophosphate-dependent phosphofructokinase. And like, for example, from the genomic data, we understood that Haemophilus influenza was predicted to have ATP-dependent phosphofructokinase. And we compared it, and we have an ability to compare across the board with all 300 different organisms for which we have metabolic reconstruction. And Treponema pallidum, which causes syphilis, it has pyrophosphate dependent phosphofructokinase. So you can start to see the patterns. And if you will go systematically through the biological system, you can actually understand quite a bit about its functionality. So we also sometimes, when you're looking at the pathway, you see that some enzymes, there are gaps. Like, for example, we didn't find phosphofructokinase in one strain of Haemophilus. So what we are doing is we offering the user to select the phylogenetically close phosphofructokinase, blast it against genome, and voila, we found the good candidate for this function. So this is a good hypothesis for experimentalists to go ahead and actually verify that this is really it. We can do it from KEGG. We can compare different strains, like what is the difference between different strains. And what is important is to have different ways to look at the pathways, different ways to look at the sequences. And this is a phenotypic point of view, like show me how it's done by thermophiles, or show me how it's done by pathogens, or show me how it's done by, for example, alpha proteobacteria. So that's what we are trying to do. We have created, but what we started to think about is, okay, life is a continuum. So that's why everybody is so similar to each other, similar but not identical. So, and like, for example, alcohol dehydrogenase, it's implemented differently in different organisms and in different domains. In some organisms, it's one subunit. In some organisms, it's two subunits. In some organisms, it's ATP dependent. In some organisms, it uses, so there could be different variations of the same functions against the genomes. And the amounts of sequence data now allow to look at these variations. And that's why we created the rules-based clustering algorithm, which allows to look at these variations. Why is it important to identify these taxonomic and phenotypic variations of functions? First, for identification of the protein and high throughput analysis, but what is interesting is diagnostics. If you want to, for example, there is a biodefense problem. You want to identify organisms without sequencing really fast. So in this case, if you have an array, a library of the taxonomy-specific enzymes, oligonucleotides, you can use it for identification of the organisms in vitro. It will be interesting for biotechnology because if you look at the thermostable variation of enzyme versus non-thermostable, then it will be also interesting for biotechnology. It's very interesting for interpretation of metagenomes, and Nikos is doing the metagenome analysis. So it's useful to do accurate metabolic reconstructions and annotate the organism-specific pathways. So the idea is to identify what is the same and what is different and how it affects function. We used this for annotation of the metagenome from Hanford site. Hanford site is the DOE high nuclear waste storage facility. And under this extremely high levels of radiation, tanks. One of them started to leak. And this metagenome was obtained from this population, which lives under the leaking high radiation nuclear waste in boiling nitric acid with extremely high concentrations of chromium. Every single condition in this pushes the boundaries of life as we know it way beyond our imagination. So we're analyzing these organisms, and actually it helps to identify the patterns, so the way we are doing it. How different are these taxonomic variations of enzymes? This is a small example which will show you. This is phosphofructokinase. No, this is fructose bisphosphate aldolase, another glycolytic enzymes. Yellow line is the consensus sequence for Pasteurellacea, which is Haemophilus. The blue line is the eukaryotes. The pink line is Enterobacteria, and this is the alignment as represented by Powys. So you see, this part is completely different in eukaryotes. These motifs are completely different in Pasteurellacea. Then Enterobacteria has its own unique part. So it can be played with. It can be used for different purposes, these differences. So Hanford site metagenome analysis, we talked about this one. We have actually identified over 28 species that live in these cozy conditions. So we have PATHAS database of pathogenic factors. We have brand new Centro database of signal transduction proteins, and this is how it looks like. And it's superimposed on the pathways, and you can do comparative analysis. The signal transduction systems, it's like the sensors for bacteria. And if they don't sense the sequence signal, then they can't do anything about it. So if you will identify what they see, you can infer their physiology to some extent. Target database. And this slide is unintelligible. But it's actually, I put it here just to show you the complexity of computational backend. And the system consists, that drives all this analysis, it consists of four conceptual parts. So first you need to have portal, so you can support analysis by the groups, and you can have the secure environment. So, and partitioning of the web space. Then you definitely cannot do all this analysis manually. So we're expressing our workflow pipelines in virtual data language, VDL, and Chimera system. We're interacting closely, working closely with the Globus group, Ian's Foster group, and we're expressing the workflows and checkers in meta languages. So, and also it's necessary to do that to drive processes to the grid. And we are using grid as a computational backend. Both University of Chicago and Argonne, they have very extensive supercomputing capabilities, but it's not enough because you need the reservations, and we are too lazy to wait for the reservations. So if we want to do high throughput genome analysis, we are just grabbing the last run that we had, it was 1,700 CPUs, and some of them, they were, just one second, they were in South Korea, some of them were in Europe, some of them were, we're using several different grids to run the processes. TerraGrid, the Open Science Grid, DOE Science Grid, so we're using different grids. And this is another way how to look at the system. So we have user-integrated database, and we have knowledge base from the public resources. We have user annotations, and we have workflow managers, and we have user interfaces. So, and all of them, they, we are trying to automate as possible, automate the update cycle, because it's very large amounts of information. And this is the schema from the last run, the resources that we took. We are mostly using Open Science Grid, which was created by the physicists. Physicists somehow can find common language. It never happens in biology. Biology, it's difficult to find common language. And they will complain about common language. So that's what drives, these are different technologies that drive all this automated workflows behind the scenes. So everything is pink and rosy, and we are rolling ahead, but we do have problems. So the first problem that we have, what is happening in biology? I wanted to give you a slide, but unfortunately, somehow it slipped out. The number of the biological ontologies that are currently being developed or just in a state of development, and I'm personally a member of like 5 committees on the development ontologies, and in Russia, and I'm Russian as you can deduce from my accent, in Russia there is this saying which says, you are friends against whom? And this is approximately what is happening in biological ontologies, because different groups are developing ontologies, and sometimes they merge them, but sometimes it's very difficult to merge them. And I personally think that somehow it happened that ontology got into the domain of social problem. But I think it's actually a technological problem. And technological problem, I can illustrate why is it technological problem and why ontologist problem should be probably solved slightly differently. But I can be wrong. I was wrong so many times. So this is glycolysis as represented by KEG, all right? This is glycolysis as represented by EcoCyc. Please see how many, what is the difference in number of components. This is glycolysis represented by MP database. Actually, MP database has 62 different versions of glycolysis that are different in different like enzyme composition and everything else. So how people can agree in the notion of glycolysis? I don't think it will ever happen, because all these representations are absolutely valid. This is just, it's normal that people have different opinions about abstractions. And what I think, somehow people got pretty good ontologies for representing facts, like sequences or sequence features, something that is, can be interpreted unambiguously, although we biologists will always find some ambiguity in everything. But once you will go to abstractions, you will never, ever, ever find any social consensus in representation. And that's why I think that what will be the solution to this is some notion of clustering of notions, some notion of clustering of abstractions, and distances between different notions and distances between different abstractions. And everybody will make the decision how little or how far they want to go in the relaxations of the ontologies or their knowledges. So,
could you go back? Yes, there. Can you explain us a little bit the differences between these diagrams here? I'm lost a little bit here.
So what is happening is there is a metabolic pathway which is called glycolysis. This is a pathway which is how to break glucose to pyruvate. Some people think it's not to pyruvate. Some people think it's further down. Some people will include some additional reactions to this. Some people will not include the additional reaction. Like, for example, in this particular diagram, this is the all-inclusive diagram from KEG. And as you see, it's called glycolysis. It's a very popular database. They just, people using it a lot, okay? This is representation, much more reduced representation of the same pathway glycolysis. People just name it differently. Like it's glycolysis, but they include only these steps to pyruvate.
So is
and pyruvate is here.
So is this one a subset of
this one is a subset of this one. But if you will tell biologist, what is this? They will say, this is glycolysis. If you ask biologists, what is this? They will say, it's glycolysis. If you ask biologists, what is this? They will say, it's glycolysis. There are 62 different versions of glycolysis. One enzyme will be different. One step will be different. They will still recognize that this is glycolysis. It's almost like a chicken. The white chicken and the black chicken and the chicken, it's still a chicken. Everybody knows it's a chicken. But somehow we know how to cluster all these chickens in our head. But ontologists, they don't know. Like, for example, I'm a member of the BioPACS group for the development. Okay, they will say, glycolysis is this. Okay, whatever. So it's a different, like, and I'm not sure that it's possible to find a social consensus to it. I'm not sure that I think it's an intrinsic problem of the data. And the thing is that in biology, like physics, they have less problems with ontologies. And the reason why is because they have a culture of abstraction which was developed pretty well. Biologists, they don't have well-developed culture of abstractions, and that's why probably it will be very like people will say glycolysis. Do they mean this or this or this or whatever? So
they're all basically ways to get from glucose
to pyruvate.
So is it time to bring the physicist again to the picture? At least one of them helped with the DNA, right?
Absolutely, and we actually, after the science food camp that was here, we're actually starting to organize the workshop with Michael Berry, who is a quantum physicist. So we want to bring the physicist to biology. But this one, I think it's mostly the problem of like how you will name the things that don't have strict definitions. So, and there is a give them strict definitions, but how you will, like for example, this part, like glycolysis, it's a well-known pathway. How you will name this part of the network, which is not a self-sufficient pathway. It's just something. You need to formalize, in this case, you need to formalize every part of every network you can get and give it a name. Or somehow establish the connections, like, for example, this set of, let's even consider it, like, look at it as a graph. This node is most likely to be connected to this. And give me, like, the depth of the network like that. I don't know. I know the problems, I don't know the answers. But it's actually very, very strange that there are good ontologies, but somehow, like for example, high throughput integrative people like us, we are not using them because they are restrictive. We're just being good citizens, we go to all the meetings and that's about it. So, we need technologies for clustering notions, definitions, and abstractions. And they should be hierarchical to some extent, somehow. Databases. So, databases. The thing is, we're analyzing the genomes for users. And users always have one property in common. Everything is different. They always want more and different. So what they want is they want to customize the databases. They want, like, for example, we're analyzing malaria parasite genomes, and they want to import the malaria parasite genomes from the different databases. But the other people who are in Haemophilus, they can't even spell apicomplexan. So in this case, they don't care about this. And when it becomes like a Christmas tree, you have this core of integrated information, but everybody wants their own like needles, and it becomes absolutely unmanageable. And in the situation when the ontologies are not very well developed, what to do? I don't know. We're just saying, okay, we can do what we can do, but not more than that. But it's not the solution. It's not the solution in the long run. So we have this problem. The other problem is actually most of the data in biology and bioinformatics, it's in the form of the trees or networks. And we have very good relationships with Oracle people. And we're interacting quite a bit with them because we have an identity crisis for our data. It's like, is it relational database or is it like object-oriented? No, maybe let's unflatten the nested tables because they're too slow, or let's nest the tables. So we're constantly looking for some solution to the problem that actually is a real problem for us, how to represent and navigate in complex networks. And when the networks are very large, what to do with them? Because if you want to represent all of the reactions, or you want to represent all of the connections between the proteins in the protein space, you're lost. It's a hard problem. Algorithms. So let me tell you about another madness that is going on in bioinformatics. So you've seen this exponentially growing data. So what is happening? One of the major tools in bioinformatics is BLAST, which is based on pairwise comparisons between the sequences. You take the sequence and you're comparing it with all sequences in the databases. The size of the database is they're increasing, doubling or tripling every year. So what we are doing is we're very optimistically and joyfully go into the n-squared problem. Because very soon, BLAST is already starting to lose its functionality because it's, if you are getting like the BLAST results for 100 strains of the same thing, it's not informative. So you can't identify the weak signal anymore. So it's, and we are personally trying to get rid of BLAST, but it will be wonderful if somebody will get rid, please, of BLAST, because it's madness. And basically what BLAST does, it does memoryless Alzheimer-style clustering on the fly. Because it does clustering, it forgets about the results, and then it goes to the next one and does it. So it's I don't think it's an efficient way to do analysis. So, and the other thing that we absolutely need, because as we discussed before, we want to look at multidimensional patterns. We want to see how things are co-evolving in the biological systems. We need algorithms for identification of multidimensional patterns. Like, for example, how to correlate the particular type of the enzyme characteristic for a certain metabolic group with a taxonomic representation of network and to project it on the genome. How to analyze these multidimensional events that were taking place. I don't know. It's the problem between in bioinformatics is the difference in language between mathematicians and the biologists. Biologists think in pictures and complexities. And mathematicians, especially, I worked with Ross Overbeek, who was a logician at some point, and he was asking me what is true and what is not true. And what is true in biology, sometimes this, it's conditional behavior. So it's very difficult to actually explain what to do. So we need these algorithms for identification of multidimensional patterns, so we can make sense on the evolutionary events. And this is my group, and I was very, very lucky. Everybody is just unbelievably talented and creative, and really love what they're doing. And I think that this is why we are just charging ahead and doing things. And thank you very much to the Globus guys. They really helped us a lot. We couldn't do anything without them. And our managers and everybody. Thank you.